Okay, we're now in Unit 3 of Blood Bank, and in this unit, so in Unit 1, we talked about concepts relating to immunology and inheritance, kind of how the body responds to immune attacks. In Unit 2, we talked about the main blood groups that you think of when you think of blood typing. So we talked about ABO and the RH, but there are so many more blood groups than just those. So in this unit, we're going to talk about some of those other blood groups. And again, we're not covering all of them. We're just covering the major ones that you're going to encounter in the blood bank lab. So that's what this first lecture is going to be on. Later, we're also going to talk about detection of antibodies. So if a patient has antibodies, how do we find and identify those? And then later, we're going to talk about autoantibodies and autoimmune hemolytic anemia. Okay, so the first blood group, and there is in Moodle, there is kind of a summary chart that has all of these blood groups that we're getting ready to talk about. It summarizes all of those blood groups and some of the major characteristics. So I would highly recommend accessing that chart, especially when you're studying. Okay, so the first blood group is known as KALE, comprised of 36 red cell antigens, following numeric and alphabetic terminology. So there's presence of detectable antigens on the fetal red cells. So these KALE antigens are detectable on babies. So, and because of that, antibodies with this antigen with the KALE blood group system are associated with hemolytic disease of the fetus and newborn. So some of the antigens, so JSA is, and I'm not sure how they name that, how JSA makes, how you make that connection with KALE, but this JSA is an antigen in the KALE blood group system. So JSA is dominant in the Black population. So, and that means that so your antibody to JSB is seen more in the Black population, because if they make A, they can't, if they have the antigen A, they can't make the antibody to A. So that means they would be able to have the JSB antibody. Other antigens, so the little k, KPB, JSB, are considered high frequency. So that means a lot of people have those antigens on their red cells. So because of that, antibodies to those antigens are very rare. And there's several of these antigens that we will talk about that are considered high frequency, meaning a large part of the population will have that antigen on their red cell. And remember, if you have the antigen, you cannot make the antibody to that antigen. Almost 100% of the population is positive for the little k antigen. So incidence of anti-little k is very rare. And you can consider that information when you're if you've discovered that a patient has an antibody that's reacting in your testing and you have to find out what that antibody is. Information like this, like the frequency of an antigen, and especially frequency amongst different populations or subgroups of people or races, that can help you determine, is this patient even capable, or is it likely that this patient has developed this antibody? Okay, antibodies in the KALE blood group system are considered clinically significant. These antibodies are going to be of the IgG class, so they're produced following an antigen exposure, and remember that has to happen through either transfusion or pregnancy. Antibodies in the KALE blood group system can cause hemolytic transfusion reactions, and antibodies in this blood group system will agglutinate optimally in the IAT reaction phase of your testing. Antibodies in this blood group system usually do not bind complement. the optimal in vitro binding temperature is at 37 degrees at AHG. So this would be when we're doing our testing. It's going to require an incubation phase and addition of the AHG reagent to see that optimal reactivity. Enzymes, so we talked about some different enzymes that can be used in our testing to either help enhance or destroy certain reactions so that we can kind of rule out certain antibody classes and help us kind of hone in on the antibody identification. So within this blood group system, enzymes do not affect the reactions. So either papain or physin, those are some of the enzymes that we talked about. However, some antibodies can show dosage. And I think we talk about dosage a little bit more later on, but it's just the idea that you're going to see stronger reactivity with homozygous versus heterozygous expression. Okay, the KX antigen and the McLeod phenotype. So the KX antigen, even though it is independent of the other KEL antigens, it still has a phenotypic relationship with the KEL blood group system. So in this phenomenon called the McLeod phenotype, you've got the XK gene, so it's assigned to the X chromosome. It's not inherited. So the KX antigen is not expressed on the red cells, and you'll have reduced expression of the other KEL antigens because of this. So this is associated with a red cell abnormality known as the McLeod phenotype, where there are morphologic and functional abnormalities of the red cell. So this is very rare. It almost exclusively is going to occur in males, and it'll be characterized by reduced red cell survival, and acanthocytosis can be seen on the differential. Okay, the next blood group system is called Duffy, and it's abbreviated FY. There are 5 antigens associated with the Duffy blood group system. So you've got Duffy A, Duffy B, 3, 5, and 6. So your frequency for whites and blacks. So whites, you can see Duffy A and B positive is the most common frequency. A negative, B positive would then be next, and then A positive, B negative is the least frequent in whites. And then in blacks in the US, A negative, B negative is the most common phenotype. So kind of the opposite of the whites. So again, that can help when you're trying to identify antibodies that the patient potentially has. And then Duffy A negative, B positive is next frequency for blacks. Again, antibodies in this blood group system are of the IgG class. Agglutination reactions are going to be best in the IAT phase of testing, so they are considered clinically significant. And once again, stimulated by transfusion or pregnancy. So the patient has to be exposed to the foreign antigen in some way through transfusion or pregnancy in order for them to start producing those antibodies. This one can cause delayed hemolytic transfusion reactions. Antigens FA and FB are going to react optimally at 37 degrees Celsius at AHG. They can demonstrate dosage as well, but these guys are destroyed by enzyme treatment. So if you got it down to, say, you're trying to identify an antibody that a patient has, and you've got it narrowed down to Duffy and, well, let's say KEL. One of those things that you can do is introduce an enzyme, and if you know that the enzymes will destroy Duffy antibodies, but they're not going to affect KEL antibodies, then you can add that enzyme, look for the reaction, and then determine which one that you're dealing with from that. Duffy A negative, B negative phenotype. So people who are Duffy A negative, Duffy B negative, confers resistance to Plasmodium knowlesi and Plasmodium vivax. So these are two of the parasites that are known to cause malaria, but does not confer resistance to Plasmodium falciparum. So you're going to see this phenomenon more commonly in the West African population. You're going to see this incidence of resistance. Okay, next is the KID blood group system, abbreviated as JK. So there's 3 antigens associated with this blood group system, JKA, JKB, and JK3. So the antibodies that the patient can form are going to be anti-JKA and anti-JKB. Those are going to be the most common. You don't really see JK3 that much. These antibodies are of the IgG class that occur in response to transfusion or pregnancy. Antibodies in the KID blood group system are associated with delayed transfusion reactions and extravascular hemolysis. Agglutination reactions occur best during the IAT phase of testing. And a lot of times if a patient has a KID antibody, a lot of times they will be seen, those antibodies will be seen in combination with other antibodies. So if you've identified that a patient has KID, there also may be other antibodies as well. Detection of these antibodies can be helped by using enzymes, reagents. Also LIS and PEG are some of our enhancement media that we talked about previously. So use of LIS or PEG can just enhance those reactions with the KID blood group system. Antibodies reactivity is going to decline quickly in vitro. So that's kind of one reason why it's important when you're doing your testing not to get sidetracked or pulled away, or leave a lapse in some phase of the testing, because some of the reactivity with some of these classes could start to decline. So there's also a phenotype referred to as KID null, and remember, null just means kind of the absence of. So what's happening in this phenotype is a patient or a person does not inherit either the JKA or JKB antigens, so they're negative for both, which is referred to as KID null. That phenotype is most common in Far East and Pacific Islanders, and this is what produces that unique antibody JK3. So we mentioned up here that was one of the antigens in this blood group system, but it's not a very common antibody, and that's because it's only seen in this certain population of people. So again, your phenotype frequency, I'll let you look at that and just kind of see the differences between whites and blacks. And when you go to work in the clinical lab in blood bank, they're going to have these frequency charts on the wall in the lab. We have them on the wall in the lab in the classroom. And it goes into more frequencies than just for whites and blacks. But again, those charts are there because that can be a tool in helping you determine a possible antibody identification for a patient. Okay, next is Lutheran blood group system, abbreviated as LU. 25 antigens in this blood group system, but the 2 main antigens are Lutheran A and Lutheran B, and Lutheran A negative, B positive is the most common phenotype in most populations. This one has a unique relationship with the SE or secretor gene located on chromosome 19. And the blood bank guy has a really good video on that. I would suggest watching that. If it's not in Unit 3, I'll see if I can find it and put it there. But and I think the whole video is about secretor genes. Okay, so the antibodies that can be produced are anti-Lutheran A and anti-Lutheran B. with anti-Lutheran A. This one is one of those kind of weird ones that may be present without immune cell stimulation. So what does immune cell stimulation mean? That means that your immune system has been stimulated by a foreign something. So that's where the transfusion or pregnancy comes in. So this can happen without that immune stimulation. There are IgM and IgG classes of anti-Lutheran A antibodies. Your optimal in vitro agglutination is going to be at room temperature, and you might see a mixed field pattern of agglutination that would be more common with an anti-Lutheran A. There's really no clinical significance to these as far as transfusion goes. You may occasionally see mild cases of hemolytic disease of the fetus and newborn. With anti-Lutheran B, this one is a pretty rare antibody that you would identify in the clinical lab because, again, it is a high incidence antigen. So if a lot of people in the population have the antigen, they cannot produce the antibody. It is an IgG class antibody. Most of your agglutination, optimal agglutination, you're going to see at the AHG phase of testing. And again, you could see some mixed field reactions with this one. This one is associated with transfusion reactions and mild hemolytic disease of the fetus and newborn. If it's associated with some type of hemolytic transfusion reaction, those reactions are usually going to be mild and delayed. But again, this is a very rare antibody because it is a high incidence antigen. Okay, Lewis blood group system is next, abbreviated as LE. 6 antigens. The main ones are going to be Lewis A and Lewis B. And those antigens are not integral to the red cell membrane, but so the ones that we've talked about previously, it's kind of like they're a part of the red cell membrane. But with Lewis, these are passively absorbed onto the cell. The primary source of Lewis antigens found in plasma is from the GI tract. So there's a relationship between these antigens and the GI tract. They're inherited at 2 independent loci on chromosome 19. So you've got Lewis A and Lewis B that are not alleles. This one is another one that's closely related to the secretor genes. So Lewis phenotype, Lewis A negative, B positive, comes from the inheritance of an LE, so inheritance of your Lewis and your secretor gene, and this phenotype produces both the Lewis A and the Lewis B antigens. So if a person has the phenotype Lewis A positive, B negative, so opposite of what we just talked about a second ago, those people are considered non-secretors. Bombay individuals, who we talked about in Unit 2, can't express the Lewis B antigen. So a person can be a non-secretor, in which case they would be identified with that little se, and still, so a person can be a non-secretor and still secrete Lewis A antigen into body fluids. The Lewis antigen found in secretions are glycoproteins. The ones that are found in plasma are glycolipids, so just a slightly different chemical structure there. Red cells absorb only the glycolipids onto the membrane. Adult red cells that have the phenotype Lewis A positive, B negative is very rare. And then just kind of as an interesting fact, during pregnancy, there's a reduction of Lewis antigens on the red cells, and mom can actually become Lewis A negative, B negative, and during that time, she can make Lewis antibodies. So it's kind of interesting. And again, I do recommend watching that blood bank guide video on how this blood group system correlates or corresponds with the secretor status. And again, I'll let you look at the phenotype distribution between whites and blacks there. Your antibodies. So you can produce anti-Lewis A or anti-Lewis B. Most Lewis antibodies are going to be of the IgM class. Glutination reactions are going to be best at initial spin and maybe 37, rarely at AHG. So, when I'm talking about the antibody class that the antigen falls into, if it's IgG, remember those are warm antibodies, so your optimal reactions are going to be at warmer temperatures. So either at 37 degrees, so during your incubation step, or during the AHG. IgM antibodies are considered cold, so most of those are going to be optimally reactive at initial spin or room temperature. And because of this, because it's an IgM, it's considered not clinically significant. Usually the IgM antibodies are going to be the ones that cause more issues during testing versus issues actually with the patient during transfusion. Enzymes do enhance the reactions with anti-Lewis B antibodies, and they can cause hemolytic transfusion reactions, but are not often associated with hemolytic disease of the fetus and newborn. Okay, antigens, I, big I, little I. So the I blood group system has one antigen, so that's the one that's named big I. So I and the big I and little I antigens have a reciprocal relationship. So the big I antigen expression is on most adult cells, not present on neonatal cells. But conversely, the little I antigen expression is very weak on most adult cells, but it's very strong on fetal and neonatal cells. So the antibodies that can be produced are anti-big I and anti-little I. So the anti-big I is a commonly encountered IgM autoantibody. And because it's IgM, you're going to see optimal reactivity at colder temperatures. So that would be 4 degrees Celsius, which is your refrigerated temperature, or at room temperature. So if you've got a patient who has an IgM autoantibody, not only are you going to see cold reactivity in the colder temperatures or colder phases of your testing, but you're also likely going to see reactivity in every cell that you test against, because it's an autoantibody. Because it's cold reacting, some pre-warm techniques can help to avoid reactivity during your testing phase. This one is not, the anti-big I is not considered clinically significant because there's no red cell destruction during transfusion or pregnancy, but it can cause problems during serologic and compatibility testing. So that's kind of what I was talking about a minute ago. A lot of times these IgM antibodies cause more issues with testing than they do with actual transfusion reactions. And one of the things that specifically autoantibodies can do is that they can mask other antibodies that could be present that could be clinically significant. So we can, you can use those pre-warm techniques to kind of help get rid of the reactivity with the IgM autoantibodies. So that if you get rid of those, then it might help you uncover some other antibodies that could be present. Enzymes can enhance the reactivity. So again, that's just another tool that you can use. There's a greater reactivity of anti-big I with cells that have a greater number of H antigens. So from our previous unit, you know that group O individuals and people who are A2 are going to have a higher number of H antigens on their red cells. So when this happens, it's considered a compound antibody. So they refer to that as anti-HI. There is a strong anti-big I association with Mycoplasma pneumoniae infections and with cold hemagglutinin disease. This is an autoimmune hemolytic anemia that's produced by an autoantibody that reacts best at colder temperatures. With anti-little I, it's associated a lot of times with infectious mononucleosis, lymphoproliferative disease, and sometimes with cold hemagglutinin disease. So you can do a cold panel to ID the antibody, again, and some of those other tools like your enzymes or your pre-warm technique to kind of get rid of the auto or the anti-I if you need to find other antibodies that are present. Okay, the P blood group system, so this includes P1PK, globicide, and then some other related antigens. This whole blood group system is not really that well understood, but they do know that there are at least 2 genes at different loci involved in the development and expression of these antigens. So because of that, these antigens cannot be considered one blood group system, but they're talked about and kind of placed together. Currently in your ISBT nomenclature, they're identified as the following. So you've got the P1, PK, and NOR are assigned to the P1 PK blood group system. P and PX2 are in the globicide blood group system. The P1 antigen is associated with hydatid cyst fluid. So this antigen can be found in secretions from those cysts, and if you haven't talked about it already in Special Micro this semester, you will. So this is fluid obtained from the infection from the dog tapeworm, Echinococcus granulosus, that causes that hydatid cyst. Landsteiner and Levine discovered the P blood group system in 1927. They did this by injecting rabbits with human red cells, and then the rabbits produced antibody anti-P. And then from that testing, they divided human blood into 2 groups. They referred to it as either P positive or P negative, and later they changed those names to P1 and P2, and then P null became little p. There is a chart in the outline that talks about the different like the P blood group summary. So it has the phenotypes, the antigens that would be present in those phenotypes, the possible antibodies that could come from those phenotypes, and then the frequency chart. So I don't think, oh yeah, I did. Yeah, I did include it here. So take a look at this, but don't spend a lot of time on it. P is not really one of those very significant systems, so we don't look at it a lot, but just glance over that chart. Okay, so your antibodies in the P blood group system, anti-P1 is common. It's naturally occurring, which means that it does not require immune stimulation. It's cold reacting, so it's an IgM. There are some rare examples that can react at body temperature, but normally the cold reacting, room temperature reacting antibodies are going to be considered clinically insignificant. Anti-PP1PK, so these individuals have a rare null phenotype that can make this antibody. It is considered clinically significant, but it is rare. Women who have this antibody can suffer from recurrent spontaneous abortion. And then transfusion recipients that have this null phenotype require donors with the little p phenotype. Auto anti-big P, which is an IgG antibody that's also known as the Donath-Landsteiner body, is associated with an immune hemolytic anemia called paroxysmal cold hemoglobinuria, or PCH, where you'll have patients that can exhibit hemolysis and hematuria on cold exposure. So these patients have to be in a warm environment at all times, because exposure to that cold can cause an exacerbation of this paroxysmal cold hemoglobinuria. And what happens is it binds to P positive, which can be either P1 or P2 red cells at lower temperatures, complement activation occurs and it produces hemolysis when the red cells are warmed to 37 degrees Celsius. This condition can appear transiently in children after they've been subjected to viral infections, and it can also be seen in adults with tertiary syphilis. It'll react weakly or not at all in routine in vitro testing and requires the Donath-Landsteiner test for confirmation. And that's what this is. So it's just kind of a series of incubation steps at different temperatures to see if hemolysis is present. Okay, skipping ahead over that other P chart that we looked at, the MNS blood group system, 49 antigens expressed primarily on the red cells. The main antigens that we'll talk about and that you'll encounter are the M, N, S, and U. So there are 2 genes that encode the MNS system antigens. They are glycophorins. They're on chromosome 4, so they're inherited as a haplotype. So one gene expresses M or N, the other codes for big S or little s. The most frequently inherited haplotype is big N little s, followed by M little s, M big S, and N big S. So the U antigen is always present when either big S or little s is inherited. Okay, antibodies that can result from this blood group system. Anti-M, of which there are both IgG and IgM forms that exist, occurs naturally. It's usually clinically insignificant. They could be potentially significant if they're reacting at AHG phase in cases where a pre-warm procedure is used. When you have an anti-M that's showing reactivity at 37 degrees Celsius, M negative units should be used for transfusion. This one, anti-M, is a rare cause of hemolytic disease of the fetus and newborn. This is this blood group system, MNS, is where we see probably the strongest incidence of dosage. And this happens because, like I mentioned previously, your agglutination reactions are going to be stronger with a double dose expression of the antigen, which is homozygous inheritance versus heterozygous inheritance. So this means that you can see, so heterozygous expression means that hetero means different, so you've got one M and one N. Homozygous expression can occur with either 2 M's and no N's, or no M's and 2 N's. So either one of these would be considered homozygous because you've got the double expression of the M and none of the N. So when that happens, if you've got inheritance of a double dose of the M antigen, you're going to see stronger reactivity in the testing because of that. Anti-N, you don't see this antibody very often. It's an IgM cold reacting antibody, so it's not usually considered clinically significant. It can be found in dialysis patients after many transfusions. Exposed to formaldehyde sterilized instruments, not clinically significant. Anti-big S, anti-little s, and anti-U. So these are clinically significant because their IgG can cause decreased red cell survival and hemolytic disease of the fetus and newborn. The U antigen is very high incidence, so it's seen in over 99% of the population. So antibodies to the U antigen or anti-U is very rare, but you might suspect an anti-U antibody if the serum from a previously transfused or pregnant person of African ethnicity contains an antibody to a high incidence antigen. U negative blood is in less than 1% of the Black population, and it's not present in white donors at all. Incidence of anti-U increases in people who are negative for both the big S and the little s antigens. This last little bit, this is a chart from your textbook, miscellaneous blood group systems. So I told you there are tons and tons of blood group systems. We only talk about the ones that are most frequently encountered, or the ones that are considered clinically significant or might cause issues with our testing. So the chart on these next 3 slides, and again, this chart is in your book. This chart goes over some of the other blood group systems, and some of the defining characteristics of those blood group systems. We are not going to go into these just because these are not something that we typically or frequently test for in the clinical lab, and you don't really see it that often. So just look over this chart and be aware that there are all these other blood group systems, but we're not really going to go into them. So that's the end of this first lecture for Unit 3. The next one will be on antibody detection.